Ok, online. Ao vivo. All right.
Agora sim, estamos ao vivo. Muito bom.
Vamos lá. Vamos começar, acho que...
Podemos iniciar então, pessoal. Muito boa tarde a todos, né? Aos que vão chegar aqui, que vão assistir depois, posteriormente, a gravação que vai ficar no nosso canal da pós. A gente está aqui hoje dando continuidade à nossa disciplina de seminários 1 do PPGECO, da UFSC. E hoje nós temos a apresentação da Alix Matthews. A Alix, ela é graduada em Ciências Ambientais pela Rhodes College no Memphis, Tennessee, e ela desenvolveu seu mestrado também em Ciências Ambientais na Arkansas State University, em Jonesboro, no Arkansas, né? Seguiu trabalhando como gerente em um laboratório em Tyler, no Texas, onde ela orientou alguns alunos também, e desenvolveu seus próprios projetos com formigas cultivadoras de fungos, né, a Attine. Atualmente, a Alix ela desenvolve seu doutorado em Biociência Molecular na Arkansas State University, em colaboração também com o doutor Fábio Akashi, aqui do nosso PPGECO, da UFSC, estudando a evolução dos ácaros de penas de aves, sua biodiversidade e a natureza simbiótica. Ok? Então, agora a gente vai iniciar a apresentação da Alix, e eu peço a todos que coloquem suas dúvidas, suas perguntas no chat, e a gente vai, ao final, fazer um bate-papo com a Alix e poder responder alguma das perguntas, ok? Alix, please welcome.
Obrigada. Thank you. I have my text in Portuguese, but I'll be speaking in English. But so my presentation is on feather mites, and sort of just, we're looking at feather mites from a number of different biological levels. And so I'll kind of go through our different levels of how we're looking at these symbionts. And symbionts are everywhere. So this is symbiosis is sort of any relationship that is between two closely interacting and distinct organisms, and they are incredibly common in our world, from the intracellular level with our mitochondria and viruses, to the individual level with sort of like our gut microbiome, to more broad scale, like with leafcutter ants or sea anemones. And symbionts themselves make up a large slice of the pie of life, so to speak, with symbiotic bacteria taking up a huge portion of the biodiversity in our natural world. But many of these symbiotic species are not described. They're not known. So there's a lot of unknown biodiversity when it comes to symbionts themselves. And the functional nature of symbiotic interactions are typically organized into these really distinct boxes, where we think of parasites or mutualists or more commensalists. But these relationships can actually exist on a continuum, and the nature of these relationships can change. And one example of this is with acacia thorn ants and their plant host. So these ants will typically protect the plant from herbivory by kind of attacking the predator. But if these ant populations become too dense, so they become sort of overabundant, the plant will. So typically it's a mutualism where it's protecting its host plant. But if the ant populations become too abundant, this relationship can shift from mutualism to antagonism, because the plant is not able to produce as much fruit. So even though that we typically think of these relationships in these individualized boxes, these relationships can change and vary depending on different contexts. And because these symbiotic organisms are interacting so closely with one another, we often see sort of this coevolution between hosts and their symbionts. And there's two sort of contrasting coevolutionary scenarios. In this case, this is the phylogeny of the host and the phylogeny of the symbiont, where we see sort of this perfect coevolution where the phylogenies are mirroring one another, and there's a single individual symbiont associated with a single individual host. This is more rare in reality than sort of this imperfect coevolutionary scenario where the phylogenies don't mirror one another, and there's a lot of variation in the interactions between hosts and their symbionts. And we, in this case, we often see a variation in host specificity as well. So we see host generalists, which are associated with multiple different hosts, and host specialists, which are symbionts that are only associated with a single host. And in order for symbionts to sort of fall into this generalist category and to shift from one host to another, these symbionts need the opportunity to switch hosts, the ability to survive on that new host that they've switched to, and then the capacity to maintain that symbiosis with the new host. And I bring this up now because this presentation is kind of, this is underscored throughout the presentation between host generalists and host specialists. So something to kind of keep in mind as I'm going through these scenarios. And so birds host a variety of symbionts. These are ticks so these are ticks on a bird. So they can have ectoparasites as well as endo-blood parasites. And these on the photo here, these are feather mites. These are also ectosymbiotic arthropods. They look like specks of dirt in the field where they just kind of, if you are not familiar with them, you don't really notice them, but they just look like small little specks there, but they're actually these really beautiful and interesting organisms. And I have a video of these are moving along a feather, kind of in between the barbules here. You can see the rachis in the middle, and they're just tucked in between the barbules. And these are actually really active and relatively quick organisms that are existing on a feather. So a bird itself is kind of an ecosystem. An individual bird can be considered an ecosystem in itself, as it is hosting all of these different symbionts on top of its body and within its body as well. So this is just sort of to bring these organisms to life. And feather mites are model symbionts, so they are really common on birds. They are found on almost every order of birds. They are permanent ectosymbionts, so they have to live on their host, and they're transmitted typically vertically from the adults to the offspring, usually at the nest. But there is some evidence for horizontal transmission as well between species. But despite these being really, really, really common ectosymbionts on avian hosts, less than 20% of their biodiversity is known. So there's a lot of unknown biodiversity that exists across species of birds. And these mites are morphologically and biologically adapted to living on feathers. So this is a close-up of an individual mite. They have these small feet pads called ambulacra, which allow them to hold fast onto the feathers and prevent them from being flung off during flight. And their diet also consists of fungus and bacteria and oil that the birds have on their feathers as well. So they're really specialized for living on these feathers. And because they're so specialized to birds, the presumption has always been that there's this high degree of coevolution between birds and their feather mites. So sort of that mirrored phylogeny, that perfect coevolution, that perfect one-to-one coevolution, and high host specificity between mites and birds. But there's a lot of evidence, accumulating evidence, that I'll be showing you some of it today, that is not necessarily the case. But the influence that mites have on birds, so the effect that they have, is not well understood. So whether they are functionally parasites or mutualists or commensals or somewhere in between, or shift between those categories, that's really poorly understood as well, or poorly agreed upon in the literature. And the host species that I'm focusing on are warblers. And so these are neotropical migratory songbirds in the family Parulidae. There are 111 species within this family, and 49 of those breed in North America. And they had these 111 species sort of had an explosive radiation 8 to 12 million years ago that has allowed them to diversify into these really beautiful, colorful, ecologically diverse, behaviorally diverse, morphologically diverse 111 species. And they have variation in their ecology as well as their distributions. And so some of their distributions, these are range maps, where some of their distributions are all across North America. The pink is their breeding locations, the yellow is where they migrate, and the blue is where they spend the non-breeding season. And so some of their breeding sites in this pink, they are hugely wide, while some other species have really restricted breeding locations in really specific habitats. So there's a lot of variation ecologically, behaviorally, evolutionarily within this family. And we know they have feather mites, and there's a lot of diversity in terms of how many mites they have, what species of mites they have, from what we've learned so far. So the eco-evolutionary interactions are very diverse, but there's a lot that is unknown about the mites that live on these hosts. And so some of our broad, big questions that we have are related to the evolution of mites, the biodiversity of mites, and that symbiotic nature. And so I'll kind of go through the different levels here. So our first question related to evolution is what ecological and evolutionary factors mediate the evolution of mites at the species level as well as at the population level. And then related to that is what species of feather mites exist on these hosts, and what is sort of their hosts, the associations that they have with their warbler hosts. So that's related to biodiversity. And then in terms of their symbiotic nature, what impact do these mites have on hosts at the individual level? So we're kind of going from a broad species level to a population level to an individual level understanding of feather mites. And I will start with the broadest level, so the species level. So this was some of my work that I did as a master's student, where we sort of just looked at a wide variety of warbler hosts. We had 13 species of warbler. This is the just photos of the diversity. These were in all within Parulidae, but different genera. And we also had one species of Icteridae. This is a new family within the last couple of years. This species was thought to be a warbler, but it was kind of like a weird outlier warbler. And then it was eventually placed in its own family outside of Parulidae, but we included it in the study as well. And so to collect these mites, we were able to collect some mites from window collision birds. So during migration, some birds will unfortunately hit windows and die, but we are able to use those specimens. We were able to use some of those specimens for our feather mite research if we went and picked those up. But in other cases, we go out and catch these birds in mist nets. That's what this kind of fine fine netting is here. It's kind of like a volleyball net, and you set it up, and the birds will fly into it, and then we go safely remove them, and we're looking for these mites here. This is an up close image of mites on a feather, and again, they kind of look like specks of dust, but they are actual living, really interesting organisms. And so this is sort of how we collected these mites. We just pull a feather that harbor mites and put them in an individual envelope and bring them back to the lab for processing. And I'll kind of just jump straight to the results of this study. So these are 2 phylogenies. This is what's called a tanglegram. You'll see a couple of these. I have a couple of these to show. But we have the phylogeny of the hosts on the left here and the phylogeny of the mites on the right. And all of these data here are showing the cytochrome oxidase subunit 1, the CO1 barcoding gene. And then the lines in between are just the associations between the mites and the hosts. And some of the things that's really stuck out to us from this phylogeny, or this tanglegram, is that some species of mites were found on a lot of different species of hosts. Like in this case, we have Amerodactys ischiros, which was found in this study on 6 different hosts across 2 different genera, the Cetophaga as well as the Vermivora. While in this case with the Amerodactys protonataria, it was found only on a single host. So we were starting to kind of see patterns of differences in host specificity, and that these mites are not perfectly co-evolving with their hosts. There's not sort of that one-to-one mirrored coevolution, which was really interesting. And we were also, from this work, we were also able to describe with Fabio 4 new species of Amerodactys mites, and I've denoted those with these stars. So we had Amerodactys striatominos, ischiros, protonataria, and jonesborensis. And jonesborensis, as we mentioned before, this is where Arkansas State University, the university that I am at, that's where we're located. And we collected these mites from a bird that we caught on our banding station on campus. And so we were wanting to pay sort of homage to Jonesboro. And these are images from our publication with Fabio, showing the morphological descriptions of, or illustrating the morphology of these mites. We have the male Amerodactys mite, jonesborensis mite here, and the female over here. And something else to kind of note from these images is these images of mites is that in some cases the mites are really congregated and really abundant across the feathers, but in this case with Amerodactys jonesborensis, they're more dispersed across the feather and not as high of abundance. And these are the 2 hosts. This is the Parkesia modicea and noveboracensis, the 2 hosts that this mite is found on so far. And so from this work, what we decided to do is we want to just understand more. We want to do more comprehensive. We wanted to do more comprehensive sampling of these mites on these hosts, since we only looked at 13 species of hosts and were able to find 4 new species of mites. So there's we were only looking at 13 species before, but we wanted to expand that to all the warblers, all the parulids that breed in North America, that we have access to. And so we sampled from 45 out of the 49 species of warbler that are found breeding in North America. We collected from multiple individuals and locations for each species if they had sort of a broad breeding range. And 38 of those 45 species had mites, and so there was a prevalence of 84%. And the sort of the composition of the mites that they had, 32 of those 38 host species had Amerodactys and Tyrannodactys mites, while 21 of the 38 had another genus, Trusardia. So there's sort of 3 different genera that we're talking about in terms genera of these mites. And what we thought was something that we found to be really interesting, the 7 species that did not have any mites. This is Cardellina rubifrons, red-faced warbler. This was one of the 7 species without mites, and the other 6 were also all southwestern species. So this is in the United States, the Southwest is more dry. It's more like desert, not as humid. The climate is really different, and we found that to be really interesting. That all the host species that didn't have mites were all found in sort of these desert type environments. So that's kind of just a side note, but that's something that came up with our sampling. So we collected all of these mites, and this is sort of just a quick schematic of how we process the samples. So we isolated DNA from pools of individuals, from all the mites that were found on a single feather. And we prepared libraries for whole genome sequencing, and then we conducted our data analyses. And for this particular project, we recovered 11 of the 13 protein-coding mitochondrial genes to conduct this particular project, looking at the species-level coevolution. And what we have here, this is another tanglegram with the hosts here on the left and the mites here on the right. And something that, again, sort of popped out to us is that the same species as Amarodectes protonataria, despite us looking for it across multiple or sampling from multiple hosts, we still only found it associated with the single Protonataria acitria. So this mite seems to be really specific to this host, while again, this Ischiros mite was found on 12 different hosts in this case, but it has been associated from our sampling, but it has been associated with at least 17 other hosts. So we've got this really dichotomous host specificity even within the single genus of mites. And we were, from my time with Fabio, we were able to discover another new species here. And then this is one of the this is the Protonataria acitria that we kind of focus the next step on, as well as Cetophagus cerulea, this host, which is again something else that we focus on as we're continuing on with our research. But I did want to point out the Trusardia mite. So this is I spent pretty much the month of March in Florianopolis with Fabio, looking at the morphology, taxonomy morphology of these Trusardia mites. And so these mites, this genus is found on the dorsal side, so the top end of feathers. This is looking through, this image is of a bird with these Trusardia mites, and we're looking through the ventral side, and you can see that these sort of round specks look a little different than the previous photos I've been showing you. And this is because they are on the dorsal side of the feather. And this is an SEM image of them also on the dorsal side of the feather. So they're kind of an interesting group of mites that's a little bit different than the ones that we are working with. But we worked on the morphological taxonomy of these mites at UFSC from our 21 perulids that had this host, the 21 hosts that had this genus of mite. And our next step is to look at the molecular taxonomy as well, to help support our morphological analyses of these specimens. And so moving on to sort of the population level, again, we are interested in thinking about those differences in host specificity even within one genus. So we our two sort of dichotomous species here are this Amerodactys protonotariya, which is a specialist on prothonotary warblers here, Protonotariya citria, compared to Amerodactys ischios, which is found on those 17 other hosts. But we focused our efforts on Pseudophagus cerulea as a target host species for this mite. And so sort of our questions related to the population level is, does this difference in host specificity, is that reflected in their population genetic structure? Can we start to see the impact of host specificity, not just at the species level, but also at more the population level? So we're looking at both host specificity and sort of biogeography too, to see if there's any biogeographic patterns of population genetic structure. And these are range maps for these hosts. So the pink is the breeding grounds, the yellow is the migration, and the blue is the non-breeding grounds. And we collected mites from five different locations and 6 individual hosts from each of these locations for both species. And another kind of aspect of this is not only looking at just the mite population structure, but also potentially using this information to better understand the population structure or population dynamics of the hosts themselves as independent lines of evolution, or independent lines of evidence to better understand the population dynamics, like their migratory connectivity or their population structure as well. So using both the mite population genetics to understand the hosts, in addition to understanding their own population genetics. And so in order to do this, we conducted another sort of pooled sequencing experiment where we went out, caught these birds, pulled a feather that harbored a number of mites, and then we extracted DNA from a pool of all of the mites that came from these feathers, up to 100 mites from each feather if it was possible. And so this is what mites look like at the bottom of a small microcentrifuge tube, a collection of mites. And then we did we did pretty much an identical sort of processing of the samples, where we, again, isolated DNA from these pools, prepared libraries, conducted whole genome sequencing, and then moved on to our data analyses. And all of our sequencing so far has been Illumina short read based. In terms of our data analysis, what we've done so far is targeted around 800 nuclear genes. These are orthologous genes based on all acari, but our next step is looking at the entire genome of these mites. And so just to give you a really fast overview of the bioinformatic methods for our population genetic analyses. So we took our raw sequencing reads, our raw short Illumina reads, cleaned them up slightly, and built sort of this core gene reference, which is like our 800 nuclear genes, to use as a reference to call variants, the SNPs. Excuse me. And we used GATK, BCF tools, and BWA to call SNPs. Instead of the core gene, like the core 800 gene reference, we're also building a whole genome assembly to call SNPs as well. So we're getting a full representation of the genome from that. But based on our results so far, we have for the specialist mite here, we see that the population genetics are the populations are sort of dividing by location. So each color is a different location. And for the specialist mite, we see that there is this population structure by location, while the generalist mite are sort of all mixed together, kind of indicating what we are sort of expecting with a host generalist mite. There seems to be a lot more gene flow between these populations than with the specialist mite. But one of the questions that sort of overlaps the species level and the population level questions is, why do we see so much variation? What's the underlying cause of all this variation in host specificity that we're detecting at both the population level and the species level? And again, we sort of have this huge web of interactions between hosts and their mites, with some species being associated with a single host, like this Amaroctictis protonataria, with others being associated with multiple different hosts. So what's the mechanism underlying that? And to bring back these sort of criteria to switch hosts, the first criteria that symbionts need to meet is they have to have the opportunity to switch hosts. And one factor that seems to be kind of something that we've noticed with the particular host that certain mites have in common. So this is a really good example of this. This Amaroctictis cereus, this is found on these 3 different species of hosts, which are not really all of that closely related. There's 3 different genera that these hosts are in, but they all share the same mite. So they're not necessarily super closely related, but all 3 of these hosts are ground nesting species. And so if they are all nesting on the ground, potentially competing for nesting locations, maybe even getting into fights with one another on the ground for nesting sites, that would be a potential opportunity for mites to switch from one host to another if they're actually in physical contact with one another. So this is just a single example of why there could be such variation in host specificity in terms of how they're switching, how they have the opportunity to switch hosts. There's a number of other factors that could give mites an opportunity to switch hosts, such as distribution during the non-breeding seasons, whether or not they are parasitized by brood parasites, if any of these species hybridize with one another during the breeding season. So there's a lot of other factors that we're really trying to tease apart. But the nesting location is something that does kind of jump out right away. So not just host-centric factors, but also there are mite-centric factors that could allow symbionts to meet these other 2 criteria. So their ability to survive on a new host, and then their capacity to maintain that symbiosis across different hosts. And so we conducted a study looking at, again, this generalist mite. This is the Amaroctictis ischios, and our specialist mite here, the Amaroctictis protonataria. And we looked at survival across time of the mites. So we pulled a feather, put it in a petri dish, and every day we counted how many mites were living versus how many were dead. And the generalist mite actually lived longer than the specialist mite. And so that kind of meets that first criteria in terms of moving from one host to another. They have the ability to survive on a new host. And if they're able to survive for just a little bit longer to encounter a new host, that could allow them to sort of fall into that generalist lifestyle. On the other hand, we also looked at dispersal between these generalist mites and specialist mites, and we found, interestingly, that the specialist mite dispersed more rapidly than the host. The specialist mite dispersed more rapidly than the generalist mite. So there may be a trade-off between survival and dispersal in terms of host specificity, and the differences between host generalists versus host specialists. And so moving on to sort of the individual level. So what kind of impact do mites have on hosts? And the metric that is used most often in terms of how mites impact hosts is the number of mites that are on a feather or are on a host. And so one of the questions that we're interested in is what factors explain that variation in abundance across hosts. So I've showed you some images of feathers that there are some with very few mites and there's some that are covered in mites. And so one of our thoughts, these are hypothetical examples of factors that could explain the variation in abundance across mites. So maybe it's their host specificity, with generalist mites having much higher abundances than specialist mites. Another idea is the age of the host. Maybe older birds have more mites than younger birds because they've had a longer time to accumulate greater or larger populations. Maybe there's a difference between the sexes of hosts, so maybe males and females differ in their mite abundance because females are typically the ones incubating at the nest, and if vertical transmission is really important for mites in terms of their life history, it would make sense that females would have more mites to transmit more to their offspring. And then the second kind of side of this is, okay, there may be a difference in abundance of mites, but how does the difference in abundance impact the health of a bird? So there's a number of different health metrics that we can look at to see if there's any correlation between the abundance and the condition of the bird. And if there's any evolutionary or ecological context that could be important to that. So, for example, if the health of a bird increases with the abundance of mites, we may think there may be support for them to be mutualists. While if the health of a bird or if the abundance of mites is negatively correlated with the health of a bird, that might mean that they are actually parasitic. So trying to understand the impact of mites on an individual is the next level of our understanding of feather mites. And we do see that abundance does differ by host species. So this was in our previous study, we looked at the abundance of mites on prothonotary warbler hosts. This again is our mite, our host specialist mite, and it had these hosts had much fewer mites than Cetophagus ceruleus, which is the generalist, harbors the generalist mite. And so we start to see that there are differences between, we were starting to see that there are differences between species, host species. And so we expanded that study to look at a number of other hosts, purulid hosts, and something else that sort of popped out interesting to us, that the generalist mites or hosts that harbor generalist mites tend to have higher abundances of mites than specialist mites. This was a different genus that we were looking at. So, but within the Amerodectes genus, this tends to be the pattern, which is pretty interesting. And so what we've also done is looked at how that abundance differs or is correlated with, is related to a number of different health metrics. And so we looked at a number of different species again, and we have a metric of health here, which is, in this case, it's body condition, and then abundance of mites along the x-axis. And it's also separated by precipitation. So the sort of the environmental context of that these hosts are existing in. And so again, abundance is unrelated to most health metrics that we've looked at. We've looked at body condition, nesting success, return rate, so if survival throughout the year, apparent annual survival. But when you start to tease apart the impact of the environmental context, there seems to be an interesting sort of context-dependent interaction where under high precipitation environments, we see that there is a negative correlation between body condition and the abundance of mites, while under low precipitation context, environmental context, we see that there's a positive relationship. And so the relationship may seem parasitic under one environmental context, but it may seem mutualistic under another. And so that might be part of the reason why there has been so many conflicting results in terms of the impact of mites on hosts, because it may really depend on the environmental context that these hosts are living in. And so in order to potentially better tease apart the impact of mites on hosts, since we know that there's sort of this context-dependent interaction, our next step is to kind of conduct an experiment in the field. And last year, what we did, we worked with Protonotaria citria hosts. This year, we're also adding the Cetophagus cerulean to look again at the impact of host specialist versus host generalist on hosts. But what we do, we go out and catch these birds in mist nets again, and we put these individual colored leg bands and this is a governmental band that's got a unique number on it so that we can identify the individual. And all of these color combinations are unique as well. So we can just look in our binoculars and see we can identify that individual based on the combination of leg color bands. But what we did, we caught these birds, put leg bands on them, counted the mites, the abundance of mites on the individuals, and then we used a cotton swab to remove all the mites from half of the individuals that we caught, let them go, and then a year later, so that's right now, actually we're going out in the field. We started last week. We go out and we're looking for the birds that we banded last year, and our plan is to recapture these individual birds, recount their mites, and then try to answer the question look at the experimental treatment and say, did the experimental treatment, the removal of mites, did that impact their likelihood to come back the following year, either positively or negatively? And do we see any differences between the impact of host specialist mites versus host generalist mites? And just based on our field work last week, so we caught 20 birds last year. We're going to catch another 20 this year for a total of 40. But from the 20 that we banded last year, we removed mites from 10 of them, and we had 10 as our control species or control individuals. Of those 20, 12 have returned. So I don't know. I haven't looked at the composition, whether it's 10 individuals that we removed mites from and 2 that we didn't, or if it's flip-flopped, or if it's 6 and 6. I'm not going to look at those data until we're finished with the field season. So it's really exciting that 12 of the 20 came back, but we'll see if the impact of mites, we'll see how the impact of our experimental treatment, what happened to those other birds. And so something else that we are interested in at the individual level in terms of the impact of mites on hosts is if they may potentially be acting as cleaners or potentially cleaning feathers while they're just existing and eating. So one of the things we're looking at is the dietary selection of mites. So whether they prefer, I've mentioned that they eat oil, Europygial oil that exists on feathers, but they also are known to eat bacteria and fungi that also exist on feathers as well. And so our question is, well, we know they eat these things, but do they Do they prefer certain types of bacteria, certain types of fungi? Do they avoid other types of fungi that exist on feathers or bacteria? Do they have any dietary selection, or are they just sort of grazing upon whatever comes to them? So our project here is to look at their dietary selection based on the abundance of mites as well. And so this is a hypothetical graph here, but it kind of illustrates the idea. So these mites may feed upon harmful bacteria that exist on feathers and or potentially beneficial bacteria that exist on feathers. In some contexts, they might prefer to eat sort of these harmful bacteria at sort of low population abundances, while at the same time sort of avoiding these beneficial bacteria. But if the abundance of the population on the host becomes so great that they run out of resources, they have no other option but to switch their consumption and have to eat the beneficial bacteria as opposed to the harmful bacteria. And so under sort of these low abundance mite conditions, we may presume that they are acting as mutualists, acting as cleaners. But if at these really high density situations, where we see that they're eating sort of these beneficial bacteria, that may allow us to say, oh, actually they are acting as parasites because they're eating these sort of they may be eating bacteria that improves the structure of a feather as opposed to bacteria that degrades feathers. And so there may be a sort of context-dependent switch based on the density of mites that exist on feathers. And so what we're doing to look at that is conducting a microbial analysis of feathers as well as mite intestinal contents. And we are working again with the prothonotary warbler, Protonotaria citria. These species, as you can, I put this picture here to show that these are cavity nesting species. So they're one of two cavity nesting warblers within Parulidae. And so they also live in bottomland hardwood forests where it's really humid and moist and hot as well. And so they're kind of acting as a natural petri dish is sort of what we've deemed them anyway so they're a really good species to work to conduct these microbiome analyses. But what we did, we collected 30 feathers harboring mites, as well as some control feathers, and took photos to count these macro photos to count the individual mites and to conduct the abundance calculations. And what we just did this last week, we submitted our samples for 16S for bacteria sequences and ITS sequencing. So we're doing sort of meta barcoding in this case of both the feathers by themselves as well as the mites. So basically what they ate, we're looking at their stomach contents, what they actually ingested versus what they had available on the feather to eat, to better understand what their dietary selection is. And so the idea is to look at the proportion of microbes differing between the inside of the guts versus the inside, like what they ingested versus what they had available on the feather. And if there was if we find to be that there's a significant difference between these two groups, what they ate versus what they had available, we'll consider that as sort of a selection, either for or against certain microbes. And so maybe they prefer to eat the feather degrading microbes and they avoid the beneficial feather structure microbes. But we'll regress that across the abundance of mites to look at is dietary selection density dependent for beneficial microbes or harmful microbes on these feathers. And again, I just submitted the samples last week, and so we should have the data back in a few weeks, and maybe I will be able to give you an update on that when those data come in. But sort of to bring this all back together, mites are really complex creatures, symbiotic creatures that are more than just specks of dust existing on feathers. They're really interesting organisms, and based on what we know so far in terms of the evolution at both the species level and the population level, we see that there is not that strict coevolution that's been so highly presumed for these species. It's not perfectly, they haven't perfectly co-evolved with their hosts. There's potentially a lot of host switching going on that could be a result of mite-centric factors like their survival and dispersal, or host-centric factors like the nesting ecology of the mites. But we also see that the differences in host specificity exist both at the species level, and we're seeing a reflection of that at the population level. So there is some the host specificity is impacting both the population genetics as well as the species genetics. And then in terms of the biodiversity, we do see that there are a lot of unknown associations between mites and their hosts, as well as unknown biodiversity. So unknown species that we want to combine both morphological analyses and molecular analyses to better understand their evolution and taxonomy. And then the impact of mites on sort of the symbiotic nature, the impact of mites on the individual level seems to be really context dependent. It could be related to, so in terms of the environment that mites and their host exist in, the impact of mites may shift depending on, say, precipitation or humidity. And host specificity may impact the symbiotic nature. So maybe host-specific mites have a different impact on hosts than host generalist mites. But we're conducting a number of experiments to better understand those differences in terms of the nature of the symbiotic interaction. And so I would like to thank my advisors and my committee members, as well as all the funding sources that have allowed me to do this research, and my lab mates and undergraduate assistants and collaborators, and you all for inviting me to give this presentation. And I've put my contact information below as well. And I will take any questions at this time. So thank you.
Wow. Congratulations, Alix. And thank you very much. It's a very complex work. You work in various levels. You use a series of analytical tools, and it's just amazing. You know, I work with fish, so it's a completely different environment for me. And I am really amazed with your talk, you know, really, really interesting work. I would ask you one question about how do you count the abundance, but I saw you work with photography, right? I also work with photography, and that really called my attention, and I think it must be a great tool for you to help you counting the abundance of the mites.
Yes, yeah.
And could you talk a little bit more about that while we are getting more questions from the students in the audience here?
Yeah, so the abundance question is really interesting because something that we've just learned recently is that mites do sort of move around during the day and throughout the night. And so what we do is catch these birds during the day. We will we'll hold the feathers up and take these really sort of macro lens photos of mites on the feathers, and we're able to import these into a computer and zoom in and then count the individual mites, like with a clicker, one by one, so that we get an accurate abundance measurement for each individual, and we will look at every flight feather. So we look at primary feathers, secondary flight feathers, as well as tail feathers, to conduct our abundance measure, to get our abundance measurement. What we're also working on is kind of doing like a scoring system for abundance. Since we've been working with some of these hosts for quite some time, we're really familiar with how many mites is a low level of abundance versus a high level of abundance. And so we're also giving these sort of just a score of this is a medium abundance, a low abundance, or a high abundance of mites, and putting that into our analyses as well. So yeah, but it is a tedious thing to count these individual mites, but luckily we have really nice photos to help us do that.
Great. I can imagine how hard it is to get nice and sharp pictures.
There's a lot of blurry photos in our data set right now that we end up filtering those out and having to retake them. But yeah.
All right. Thank you. Okay, I'll talk to you later on about the pictures. I'm really interested in that. So okay. I'll get some questions from the audience here. Professor Selvino asked, what characteristics have you considered for your experiment of removing the mites?
In terms, which characteristics?
Selvino, could you be more specific in this? I mean, I don't know, the age of the birds or color, whatever.
Yeah, so we take a number of kind of basic demographic data, and morphological data from hosts. So we measure their wing chord, so the length of their wing, as well as we identify how old they are, whether they're a second-year bird, so they hatched out last year. They were either born last year or born 2 years prior. That's the only way we can age this particular species, unless we have more data from banding data. And then we take their mass as well, so that we can calculate body condition based on the wing chord versus the mass.
Okay. All right. About the body condition, I had one question also. Mm-hmm. Do you take any blood samples that you may infer anything about their healthy condition?
Yeah, that's a really good question. We don't take blood from most of these birds, or for my projects, but one of my lab mates did a similar project looking at blood, like leukocyte, white blood cell counts. So he took pictures again to estimate mite abundance, and then took blood to measure stress, levels of stress, and sort of blood cell count, sort of immunological data. He's still working through those, but there doesn't seem to be much of a relationship between, based on what he's told me, there's not much of a relationship between sort of immunological measurements and mite abundance as well. So there's a lot of evidence that kind of points that they are commensals, but we're wanting to make sure that we look from a number of different angles and consider the context to make sure that what we're calling commensals are not actually parasites under one condition and mutualists in another, and what we're seeing is sort of the in-between.
Okay. Right. Also, Patricia Serafini, she said she had one question, but in fact, she has 3 questions here for you. Yeah. So let me see. In our current scenario of extreme climate change and pollution of different environments, changes in vertebrate immunity are expected, I think.
Mm-hmm. Is it expected in different? Is that the question?
Yeah, she didn't put a question mark, but I don't know if she was.
I
introducing her question, but she has more questions here. I can read for you.
I think that they, based on, at least based on our abundance data with those, with our prevalence data based on those birds that we caught in sort of the desert environment, and we found that there were no mites. It could be either they were the environmental conditions were not conducive for those mites to live. So there is probably some relationship there with invertebrates as a whole in terms of the climate differences in climate.
Yeah, that was a quite interesting data to have no mites on that species.
Yeah.
So, do you think that that immunosuppression caused by human impacts on the environment can alter the bird's natural defenses and change the individual's relationship with feather mites with they co-evolved? They co-evolved.
Yes, yeah, I think that's another really interesting aspect, and a really interesting question, because under different conditions, under different environmental and climatic conditions, the host may not be able to either ward off other parasites or things that we do know are parasites, which could put them in poorer condition. They may not produce as much oil that is acting as a resource for the mites to eat. Or maybe they build up sort of those harmful bacteria on their feathers if they're not able to preen. So depending on whether what the mites are actually eating, there could be a huge impact of the condition of the bird immunologically, and the abundance of mites under different contexts, for sure. Yes.
All right. So, would higher abundance of feather mites in a bird be a potential indicator of lower individual immunity response as well?
Yeah, that's kind of what we are looking into, and it seems like that's not the case. That's kind of operating under the presumption that they are parasites.
Okay.
That we see like an increase in abundance and a decrease, a negative correlation between more mites and lower sort of immune function. And so if we did see that, that would lend support for them to be parasites. But it seems like, again, I haven't done any of the immunology things, but based on my lab mates' results, it doesn't seem like there's much of a relationship between immune function and abundance of mites. Again, lending support for more not for mutualism, but just commensalism.
Okay. And I have another question here. Mm-hmm. So, especially it's about the Amerodactylus ischius, right? Mm-hmm. You have different species of mites on that. Right?
Yes.
And have you tested any relationship with color, shades of light and dark, feathers, anything like that could influence their abundance, the dispersion in the bird, and about
that's a really cool question. We have only done a little bit of color analyses with this Protonotaria citria, just because another lab mate was working on color. And I had the mite abundance data, and we've like briefly looked at those results, and there's some things at least between males and females, but for this ischios, we have not looked at color based on the 17 different hosts that they're associated with. That would be the color aspect is really interesting, especially since some of these birds, like in this particular case, they're blue. And so the relationship with color would be structural instead of like carotenoid based or dietary based. But it is something that we have been kind of thinking about in terms of, are the mites actually, if they have a really high abundance of mites, can that influence the color that the birds are presenting to potential mates, just because I mean, you can see through the wings. If there's a ton of mites on there, the color that is going to be reflected is maybe distorted, just based on the fact that there's so many on the feather. But we haven't looked into that, but that is really interesting. And the hosts that are associated with these with this particular species of mite, some are blue, some are yellow, some are black. This is another host of Ishiros. So there's a huge diversity in color for that particular host or that particular mite.
Especially considering the color variation of the birds, that's must be something interesting to look at.
Yes, definitely.
So Mr. Fábio is saying here, that's great, a great talk, Alex. It's amazing how you're able to do various approaches, molecular, morphology, ecology. I'm really amazed with your work to try and answer some of the long-standing questions on feather mites biology. So as far as I can see, you're going to describe many species of mites, new to science. So that's to start our steps on putting some light on these organisms.
Yeah, the biodiversity discovery of working with an organism that so little is known about is really exciting. It's really exciting to find something, collect something that nobody else has ever seen before. I do really like that aspect of it. I feel really lucky to have like a really pretty broad sort of interdisciplinary research project where we're looking at ecology and evolution and morphology, systematics, taxonomy, biodiversity. It's pretty cool.
It's a very fat PhD, you have lots of things to talk about. It's an amazing work, yeah. So Ana Luisa Figueiredo is putting a question here. So based on the few studies we have, is it possible to infer any tendency in the relation of more mutualism or more parasitic and individual level?
Yeah, that's an interesting question. Like in terms of some individuals within a species having a parasitic relationship with their mites, with others having a mutualistic, would that be sort of the idea?
Yeah.
Yeah, I would not count it out at all. I think that there's a lot of variation in the relationship, the functional relationship between mites and their hosts, and so I wouldn't be surprised if we did see something like that. But from the literature, no, that there's I can't think of something off the top of my head that would be that would explain that, or that anybody has really looked at. So that could be an interesting angle to consider.
Right in the beginning, you were giving a talk about the perfect and imperfect coevolution. And do you have any example of the perfect coevolution that you could talk about?
Yeah, they're sort of the textbook example is with pocket gophers, and so a small mammal, and their lice. So they're ectoparasitic lice. That's sort of the textbook example of perfect coevolution, with sort of that one-to-one species, one-to-one association, and then the phylogenies pretty well mirroring one another. Okay. The thing with those pocket gophers, these mammals are underground. They're really solitary, and so there's not much opportunity for their ectosymbionts to switch from one species to another.
Okay.
Yeah.
All right. That's great.
It seems like that's the exception rather than the rule. In a lot of cases, although you would expect that perfect coevolution in a lot of systems, it's a lot more complex than that in most cases.
All right. Silvino, Fábio, any more questions you have?
No, thanks. For me, it's okay.
Fábio? And any of the students, if you feel like asking any more questions to Alex?
Well, I think one of the students asked in the beginning, one general question about how long can feather mites survive after the host dies. Alex, would you like to say something about that?
Yeah, that's. I don't have the these.
I have some data also, but I'm gonna leave you first.
Yeah, this is the data that I collected. These are the data that I collected. I don't have the scale here, but this is about 2 weeks. So most feather mites, once they're removed from their host, like in this case, they were able to live for about, the longest was like 17 days, I think, 17 or 18 days. So just a little bit longer than 2 weeks. And so if the host dies, mites are either going to die along with the host, or they have to somehow encounter another host, or disperse to a new host. So that's kind of the idea of behind this project here. But I think Fabio may have another example or another answer to this question. But it seems like without their host, they can't live all that long.
Yeah. Actually, a couple of weeks is what I have also, and I was thinking that it may have something to do with the size of the feather, because you're mostly dealing with small passerines with feathers like that, a few centimeters. And I once collected a Cariama, Ciriema, and they have really long feathers, and I had mites living after 4 or 5 weeks after I removed the feather and I stored in a plastic bag.
Mhm.
And then after 4 or 5 weeks, the mice were still alive, and I think it has to do with the uh, getting rid of the water, the humidity of the feather, and then the longer and the bigger the feather, the more humidity it can hold, and perhaps it has something to do with that.
Yeah, that's true. The humidity is a huge aspect of survival for small ectosymbiotic arthropods in general, but also for feather mites, the size of the feather could also provide additional resources, for like food resources that they can consume for a longer period of time than these small passerine mites found on really tiny feathers.
I have a very basic questions. What is the lifespan of a mite?
Yeah, that is something that a lot of people want to know.
Species. How long do they live?
Yeah, that's something that we don't know about. That's a basic biological question that we don't really have a clear answer for these mites, which is really interesting because they like without their host, like if they're removed from their host a few weeks maybe, but the lifespan on a host is completely unknown, as far as I understand. We don't know enough. We don't know a lot about their lifespan, or necessarily about their reproduction on a feather as well. We know that they produce one egg at a time, but we don't know if a female can produce multiple eggs in her lifetime, or if she dies right afterwards, or anything like that. So there's still a lot of unknowns about these mites, including how long they can live, the lifespan on a living host. It's a really good question that would probably help us understand the basic biology of these mites a lot better if we did know that information.
I think the question from Lucas Felizardo was answered already. Sorry, Lucas, for not putting your question before. Fábio remembered. Thank you, Fábio. Okay. Any more questions, people?
Can I ask you something? Have you considered measuring, getting back to counting the number of mites on the feather? Have you considered using some software to measure the area of the mites? Like, the more mites, the larger the area of them. And
yeah, I haven't considered that, but that would be really interesting. We have looked at, we've done some analyses in terms of like how they're distributed on a feather, like if they're closer to the tip of the feather or closer to the quill, and if that's symmetrical between the right wing and the left wing, the right side of the body and the left side of the body, but not necessarily the area. But that would be interesting in terms of the proportion of area that they take up on a feather, because you could extrapolate that to how much space they're using on the whole bird. That would be really interesting.
Yeah, because I was thinking that some of the males they get attached to females, or even to the immatures, and sometimes that may confuse, and you end up counting one mite or two mites, or sometimes it looks like a long row of sausages, and then you have it's difficult to count them.
Yeah, and there are some cases where, I mean, if you don't look at a ton of pictures and you don't see them under the microscope, it may be hard to count each individual one, but yes, because some do kind of look like long rows of sausages that you're like, I don't know if it's one or two, but in most cases you can at least, I can imagine that it would be a male and a female in copula that I count that as two individual mites. We're certainly missing, I mean, these are not exact abundances, because there is error in our counting, and also the time of day that we take these pictures. But these are the best estimates from our efforts.
Fábio, would you talk about the students' mobility, something like that you asked in the beginning? You asked me in the beginning.
For this project, Alix, she invited me right in the beginning to serve in her community, committee of judging, her PhD, and then I was and this committee is formed with different specialists on different areas. So I think, well, maybe Alix is more qualified to talk about that. And we have this taxonomist and a genomics person, and then the ornithologist guy, and then And what she did about a month ago, a little more than a month ago, she spent a few weeks here in Florianopolis to study morphologically some of her mites, and then after having studied molecular and doing the gene sequencing, some of these mites will be sequenced and analyzed molecularly after. Also, but Alex, if you could talk a little bit about that, this variety of people and different expertise.
Yeah. So in the U.S., it's typical to have one or two like advisors that are your kind of main advisors. And the idea for a committee is to have a group of people between like 5 and 6 people on your committee that are serving specific roles in for your dissertation projects with different expertise. So like on my whole committee is made up of like Fabio was saying, an ornithologist, that's Dr. Thom Bovis. He's a ecologist ornithologist. And so obviously I'm working with birds and I have a lot of ecological questions related to mites. And so he's serving sort of that role. Acela Attine is a genomicist, molecular biologist. And so that is, he's helping with his expertise is in terms of the molecular side of things and the genomics that I'm technology that I'm using. Fabio is obviously a feather mite expert. I'm studying mites and I'm interested in biodiversity and taxonomy, and that's sort of Fabio's role, as well as in the ecology and understanding the basic biology of these mites. Travis Marsico is like he's actually a plant biologist and ecologist and evolutionary biologist, but he's sort of part of our biodiversity understanding and describe biodiversity in these mites. And then Dr. Andrew Sweet is also kind of a mixture of an ornithologist, ecologist, evolutionary biologist, and genomicist that's helping with his expertise in those realms. So I've kind of got a pretty big, interesting group of experts that can really be critical of my projects and knowledgeable and critical and helpful with all my projects in terms of the ecology of these mites, the evolution, and their biodiversity. And that's pretty typical in the United States, having a really wide variety of people on your committee. And we have a meeting usually once a semester, so 2 or 3 times a year we have a meeting where we all get together. I give sort of an update on my progress. We go through troubleshooting problems or ideas if we need to start steering in a different direction. Just sort of an update on that. And then sort of the outline of, at least at Arkansas State, the way that we progress through the program. You have to have a proposal. So I've defended my proposal already, which is where all the committee members and the public get together and listen to something similar to this, where I give a presentation on what I'm planning to do and what I've done, and they ask questions. And then we go through a second proposal for our candidacy, which is sort of like our qualifying exam, where you write write and defend another full proposal that's not related to your dissertation project. So something outside of the scope of your dissertation, to kind of get your ideas, get your mind on science outside of your specific field. And then I did that as well. I did that earlier or last year. And then the third step is defending the actual dissertation. And so that's what I'm working towards now. But that's sort of the outline of the role of the advisors and the committee members in terms of meeting and advising, and in Fabio's case, going down to Brazil and spending 3 intensive weeks working on projects side by side, which was really, really awesome.
And before you defend your PhD thesis, you have to come back here so you present the final results, and we end up describing moments.
Yes,
you give
absolutely.
Another talk.
Definitely.
Ok, Alex. Gostaria de agradecer, então, a gente caminha para o final aqui, né? Agradecer a presença do professor Fábio Akashi, né? Então, orientador da Alix, né? Agradecer ao professor Selvino também por manter, né, e armar todo esse circo para a gente poder ter aqui a transmissão ao vivo da Alix. Essa fala da Alix vai estar no canal do PPGECO da UFSC, e essa é a nossa primeira, né, transmissão ao vivo da disciplina de Seminários 1 do primeiro semestre de 2022. Né? Espero que tenhamos aí ótimas transmissões futuramente. Agradeço aqui também a Alex. Thank you very much, Alex.
Thank you.
You're always invited to talk here, and for sure you're going to come back here, and we're going to talk more about the mites and the feathers.
Yes.
The very work you've been developing. Congratulations for your work, and hope you have enjoyed it.
Yes, thank you. Thank you so much for the invitation. This was awesome.
Ok, pessoal, muito obrigado, então. E nos encontramos aí na nossa próxima palestra, que será anunciada no canal do Instagram do curso e também por e-mail. Muito obrigado pela participação aí de todos.